Hello everyone, I'm Rishabh, and I hope everyone is able to see my screen.
Yes, sir. Good evening, sir.
Good evening. And so Today we'll be discussing about week 3, and let's get started. So, yeah, here goes the first question. Which of the following plant hormones is synthesized from amino acid precursor and has a crucial role in regulating root meristem? So the options are ABA, that is abscisic acid, auxin, gibberellic acid, or cytokinin. So can anybody take a guess?
Option, sir.
Hmm. Anybody else? So yes, the answer is auxin, and it basically is formed from tryptophan. That's a aromatic amino acid, and ABA is basically formed from beta carotene, and gibberellic acid is formed from terpenoids. So the basic precursor of gibberellic acid is geranyl, and cytokinin is formed from ATP. So it's a form of nucleotide. So let's move on to the next question. So I hope everyone is able to see the question.
Yes, sir.
Hmm. So the question states how to check whether QC, that is quiescent center, is ablated. So by ablation we mean that the cell is not working and is not present there, which is its destined location. So we can either we can do the options are by iodine test, by APTS test, using scanning electron microscopy, or we can either use this or this. So, can anybody take a guess?
Both A and A or B, sir.
Okay. Anybody else? So, yes, we can use both A and B to check whether the QC is ablated or not. And the reason is that first we have to look whether what's the basic principle of these tests. So both these tests are used to detect the presence of starch in the cell. So in as you remember in QC, when we see like the layer that is very nearest to the QC are the initials, columnar initials. So it does not have any the starch content, and as it differentiates towards the end, the starch accumulation increases. So, and if any of the QC is ablated via laser ablation or any other ablation, so these differentiated cells start accumulating starch, and that's a major um, form that we can detect whether the QC is ablated or not. So we can use either iodine test or the APTS test. So iodine test is basically used to detect starch, and it forms a complex that it like starch has two components, as you all know. First is the amylose, and the other one is the amylopectin. So amylo iodine molecules basically go and bind, like stay in the helical nature of the amylose, and it gives a bluish blue brown color to the solution when the starch is present. So what we do is we just take the sorry so we take the cell and we put iodine on that, and if that iodine changes its color to blue, that means the starch is present in that cell. And the other test is APTS test, that is aminopyrene trisulfonic acid. So it is a basically a form of an acid that reacts with the aldehyde group or the carbonyl group that specifically. So starch has two ends, that is the reducing end and the non-reducing end. So the reducing end has free carbonyl group of the glucose. So it reacts with this and it gives a green color that like absorbs at a wavelength of 500 to 530 nanometers, and it can be visualized through fluorescence or confocal microscopy. So these are the two tests that we can use to detect the presence of starch. So any doubts here?
No, sir.
Yes, who has raised a hand? Do you have any doubt? Please ask if you have any doubt. Otherwise, we'll go to the next question. Or else you can write in the chat box. Okay, then we'll move on to the next question. So, the next question is, consider the following statements regarding plant development, and these are lateral roots developed from the pericycle cells, root cap cells are mainly dead cells, lateral roots develop from the epidermal cells, or the fourth one is the radial meristems are responsible for secondary growth in roots. So, which of the following is correct? So here are the four options, and can anyone take a guess? Okay, so like the correct answer is option A. That means statement A, B, and D are correct. And how? So, like, as we know that the root is divided into different zones. So the first comes the root cap, following which it has zone of cell division, Then the zone of cell expansion or roots basically arises from the maturation zone only, and it originates from the pericycle cells, not the epidermal cells. So option C, the statement is wrong, and option A statement is correct. Followed by the root cap cells. So root cap cells are already dead cells or the differentiated cells. So it doesn't have any living cell or the cells that are actively dividing, that are not actively dividing. Okay, so option B is correct. And lastly, the radial meristems that are present. So the radial meristems are the one that increases the growth of the plant in the radial direction. That is the secondary growth. So they help in the secondary growth of roots. So statement A, B, and D are correct. Okay. So any doubts in this question?
So here the root cap cells are
Hmm.
they are mentioned, they are dead cells.
Hmm.
There are some cells are dividing, like stem cell niche cells are also dividing, no sir?
Yeah, actually the root cap region, what we call is like you can see in this region. So it's basically this region, and the adjacent region comes under the zone of cell division. So all these area that is surrounding the quiescent center that contains the stem cells that are actively dividing comes under the zone of cell division, not the root cap. So that's why we are considering root cap has dead cells only.
Okay, sir. Thank you, sir.
Okay, any more doubts? If not, then we'll move to the next question. So the next question states that there are the few statements with respect to root development in plants. So PLT, that is the plethora, is responsible for longitudinal positioning of the QC. Then the auxin maxima is established at the root apex. Endodermis and cortex layer of root arises from the same initial cell in Arabidopsis. And the last one is clevata and wuschel are important regulator of root apical meristem establishment. So we have to figure out which of the following statements are true. So can anyone take a guess?
Option B, sir.
Okay. Any more answers? So the correct answer is option B. Yes. And just a second. Yeah. So as we can see from the figure that is presented on the right side, so QC positioning is controlled by two uh, pathways. First is the plethora pathway, and the other one is the scarecrow short root pathway. So plethora maintains the longitudinal or the longitudinal positioning of the QC, and both scarecrow and short root, so they maintain the radial positioning of the QC. So first statement is correct. And auxin maxima is established at the root apex. This statement is also true because in the growing plant, auxin is maintained at high concentration in the root apex, because here there are it forms a polar gradient here. And the third option, that is endodermis and cortex layer arises from the same initial cell in the Arabidopsis. Yes. So this is the cortex endodermis initial that is adjacent to the quiescent center, and it undergoes asymmetric division and forms two cells. One is towards the quiescent center, that is remains as an initial, and the other one as two cells that is that one of them form endodermis and other form the cortex. So yes, they arise from the same initial cell. So three statements, that is A, B, C, are correct. Okay. So anybody has any doubt in this question?
So actually, I have one doubt, sir. So, auctions are also synthesized in shoot apex also, right, sir? So is that the auctions are also established in root apex also, sir?
So what's your question? I didn't get it. Can you please repeat?
Oh, yes, sir. I'm asking that auction establishment also takes place in root apex also, sir. Shoot apex also, and also in root apex, sir.
Yes.
Or only in root apex, sir?
Okay. So you are asking whether its maxima is maintained in shoot apex also, or only in root apex, right?
Right, sir.
So basically what happens is auxin is present in like more dividing cells. So where there are more dividing cells, auxin is present there. And as you have seen in the shoot meristem case, where the establishment of shoot apical meristem, so in that case what happens is some regions will have more auxin concentration and some will not. So that helps in the development of that region. So it differs from region to region. Okay. So that's the
Okay.
case.
Thank you, sir.
So any more doubts? Like in continuation to like the previous doubt, so in case of the shoot apical meristem, the auxin signaling is being like negatively regulated because they have to increase cytokinin signaling. Okay. So
Can you repeat, sir?
there is different mechanisms for every cell, because we have to the plants have to shift to different organs at different time points of their development.
Is that auction is down inhibiting the response in shoot meristem, sir? Or
Yeah, as per the figure that is shown, so yes, auxin is down regulating that.
for shoot meristems, sir?
Yeah.
Is that it is suppressing the growth of shoot development, sir?
Yes.
Okay, thank you, sir.
Okay. So let's move on to the next question. In an experiment, an inducible short root GR fusion protein. So GR basically stands for the inducible system using uh, dexamethasone, and it has been transformed into the short root mutant plants in the control plants that has without dexamethasone treatment, and we do not see an expression of cyclin D gene. However, upon dexamethasone treatment, we detect the expression of cyclin D. So the options are: upon treatment with dexamethasone, short root protein can enter inside the nucleus. So, and option B, with or without treatment with dexamethasone, short root protein can enter inside the nucleus. Or the option C, short root protein is activating the expression of cyclin D. and option D, short root protein is repressing the expression of cyclin D. So yes, the answer is A and C. So what happens is we are using a GR fusion short root protein. So the short root protein is under the influence of a promoter that activates only when dexamethanosone is present here. So now what happens is when we add dexamethanosone, so short root protein will express and will enter into the nucleus, and it's also a transcription factor, so it helps in the expression of cyclin D gene. So that is explained in the figure given below, because cyclin D is here expressed with the GFP. So when dexamethanosone is added to the system, so short root protein expresses and it enters the nucleus and helps in the expression of cyclin D gene, that is expressed with the green color. So it's a translation fusion here, cyclin D with the GFP. So yes, so options correct are first, that is A, when dexamethanosone is present, so it helps in the expression of the short root protein and it enters the nucleus and helps in the expression of cyclin D protein. So this is the question. Do anyone has any doubt in this?
Sir, actually, dexamethasone is one of the glucocorticoid like hormone, right, sir?
Yeah.
So how it has been playing a role of transferring the transcriptor factor from cytoplasm to the nucleus, sir? Is there
Okay.
a scenario chemicals will play the role naturally or?
So, this question was also like being asked in the last session also, but I could not join because of some network glitch. So yes, actually, what happens was, like, do you remember the lac operon system?
Yes, sir.
Yes. So what happens is when we have a lactose in the system, so it binds to the repressor so that the polymerase can transcribe the gene and it can express. Okay. So lactose is playing as a inducer role. Here what we are doing is we are placing a promoter that acts only when glucocorticoid is present. Okay, so when glucocorticoid will be present in the system, then only this promoter will act, or this promoter will be active. Okay, so it is not playing any metabolic role or physiological role in this system, in plant system. It is just the inducer of the gene that is being used in the system under the study. So we are expressing short root protein under the glucocorticoid receptor. So when dexamethanosone, that is a glucocorticoid, is added, so this promoter or the elements that are binding to the promoter are active. Okay, so then this protein will form, and if this protein forms, then it will enter into the nucleus and it will function in expressing cyclin D. Okay.
Yes, sir. Thank you, sir. If the plant is not treated with dexamethasone, then the short root protein will not be synthesized, sir, or it will synthesize, sir?
So here we in the particular like system we are using, so we have mutated the short root protein in the plant itself. That's why we are expressing this from outside. Okay, so if we do not have the plant may have its basal level of short root protein, but that will hamper the experiment that we are doing. So first we mutate that, and then we add this in the form of a plasmid or that, and we form a recombinant plant.
So in wild types, there will be a natural expression of shorter protein surface.
Yes.
Okay, sir. Thank you, sir.
Then we'll move on to the next question. Yeah. So the next question is, which of the following statements is true for reverse genetics analysis? So the options are identifying mutants that produce a certain phenotype. So uh, second option is mutagenizing wild type with a general mutagen EMS, mapping mutant gene, making a null mutant for a particular gene to determine its phenotype. So can anybody take a guess?
Option A, sir.
Hmm. A?
D, sir.
Hmm. Okay. Any more guesses? Okay. So the option is D. So in reverse genetics, what we do is we use different genes and mutate it to check what phenotype they are showing. So when we use a phenotype to study is its genetic makeup, then it's the forward genetics. Okay, so option D is correct. Okay, any doubt in this question? So if not, then we'll move to the next question. So during root development, the columella cells stem cells give rise to the columella as a result of asymmetric cell division. At later stage, the columella stem cells show the higher expression of a protein, and their daughter cell shows higher expression of the other protein. So here are the lists, and can anybody take a guess?
Option C, Pheasant Sombrero.
Okay, any more guesses? Hmm. Yes, so the correct answer is fez and sombrero. And what happens is this is the initial cell from which the columella lineage starts developing. So first it divides, and then what happens is the the stem cells or the initials that we are talking about retains the level of fez, and the sombrero protein inhibits its function in the differentiated columnar cells, and it retains its position in the stem cells only. So this is how it functions. So anybody has any doubt in this question? If not, then we are moving to the next question. Yes. So during auxin signaling, in the absence of auxin, the auxin response factors, that are the transcription factors that work in response to oxygen, and forms inactive heterodimers with a protein. So whereas in the presence of auxin, OXIAA proteins are targeted for destruction by an activated dash. So these are the options. Can anybody take a guess?
Option B, sir.
Okay, any more? So do the correct option is option B. And what happens is, so when oxygen is present, so there is a the oxygen response factor functions in response to oxygen only. Okay, so when oxygen is present, it activates TIR1, that's a ubiquitin ligase. So and in absence of oxygen, ox IAA, that is the negative regulator of ARF, is bound to it. Okay, so that forms a heterodimer, and when oxygen is present, so it activates TIR1, that is a ubiquitin ligase, that ubiquitinates the OX IAA. Okay, so when it ubiquitinates OX IAA, then it gets the conformation is changed, and it is released from the ARF. So now ARF can function in gene expression. Now, so this OXIAA, as it is ubiquitinated, so it's a general mechanism that everything that is polyubiquitinated in a mammalian cell or in like higher eukaryotes, it goes into degradation. So here it goes into degradation via proteasome model. So this is the general scheme how oxygen signals. Okay. So anybody has any question in this? Any doubt?
So actually, oxygen is responsible for lateral root development, right, sir?
Yes.
So generally, oxygen has been synthesized in the uh,
Hmm.
root regions, and
Hmm.
then what is happening here, sir? Actually, it has been undergoing some steps,
Hmm.
and it also undergoing some set of inhibition.
Hmm.
So and then after the inhibition, it has been giving a positive response, or the another thing is happening, sir, here.
So you are talking about the steps that are mentioned here.
Yes, sir.
Okay, so that these are the same steps that I explained. So what happens is TIR1 inhibits OX IAA, and OX IAA is an inhibitor of ARF. So ARFs cannot function when OX IAA is active, and these are active only when oxygen is not present there. Okay,
Okay.
so when oxygen will be present, then it will activate a ubiquitin ligase, that is TIR1 here, that will ubiquitinylate these OXIAA negative regulators. So after ubiquitinylation, these will go to degradation and ARFs will be active. They will like help in oxygen induced gene expression. So different genes will be expressed when oxygen will be there. And after that, what will happen, that if the plant doesn't require more of those genes that are induced by oxygen, so they activate this OXIAA that will inhibit ARF. So when oxygen will be present, this IAA will be not active. Okay, so whenever the function of that root or or that cell gets over, then oxygen will also leave that cell via the mechanisms in the plant, and that will help in the regulation of this pathway. Okay.
Yeah. Yes. Thank you, sir.
Okay. Any more doubts? If not, then we'll move on to the next question. So yeah. So this is the next question. So during lateral root formation, first the lateral root primordia are formed. So the following events occur during the process, and these are the events mentioned. So what's the correct sequence of the events mentioned? Can anybody take a guess?
Option B, sir.
Okay. Any more guesses? So if not, yes, option B is the correct answer. And how did this happen? So this is the basic formation of the lateral root primordia, and what happens is these are the pericycle cells, and first they identify which cells from the pericycle itself will form the lateral root. So these are called as the founder cells, and first this is the founder cell specification. Then, as auxin helps in formation of the lateral roots, so these cells, when they are identified that yes, these are the founder cells, then auxin concentration increases in those cells. Then what happens is, as we know that in any of the divisions that take place in the development of plant, there are asymmetric division. So then they break the symmetry, and the nucleus that is present in the center of the cells come to the end, and further these cells divide, that too with the asymmetric cell division. So here the options: first we have the specification of the founder cells, this will be the first. Then second will be the auxin response maxima, so this will be the second. Third, it will be symmetry breaking, so this will be the third. And fourth is nuclear migration, and fifth is the asymmetric cell division. Okay, so this is how the lateral root development takes place. So any doubts in this? So if not, then we'll move on to the next question. So the first asymmetric cell division in the zygote is critical for shoot apical meristem establishment, and is regulated by the short suspension, that is SSP messenger RNA. So the options are: SSP regulates YDA mediated asymmetric cell division pathway in zygote. SSP regulates VOX mediated asymmetric cell division pathway in zygote. SSP mRNA is delivered from the sperm cell and translated in the zygote. SSP gene is transcribed in the zygote and translated in the zygote. So which of the following statements is true? Can anybody take a guess?
Option 3, sir.
So option C, 1, 2, and 3 are correct. Any other guesses? So the correct answer is option C. So what happens is, the pollen, wait a minute. uh, when the pollen comes and fertilizes the zygote, it brings a short suspensor mRNA, and the mRNA is translated in the zygote, and it controls both YDA as well as the WOX pathway that helps in the asymmetric cell division, and that helps in shoot apical meristem establishment. Okay, so any doubts in this question?
Uh, actually, actually, pollen grain is from male reproductive part of the plant, sir. How it has been how the zygote of the plant has been accepting that foreign particles, sir?
So what happens is, so actually, when the like when the pollen comes and they fuse with the egg cell, they form a zygote. Okay, so during that time, this mRNA enters into the zygote. Okay,
Yes, sir.
so at that time only this mRNA enters, out not after that. During that time only this mRNA enters, and these are the epigenetic mechanisms that are maintained here, so that this mRNA do not degrade and will be expressed only when the suitable conditions that are present in the zygote help it to translate.
Okay, sir.
Hmm.
Thanks.
Okay. So any more doubts?
So again, the zygote has already fused form of male and female gametes is known as zygote, no, sir. So
Hmm.
again, the pollen is getting contact with that zygote means for what purpose it has been getting contact, sir?
Okay, so what happens is when the second male gamete, that first male gamete fuses with the egg cell. Okay, so during that time, first the nucleus fuses, then the cytoplasm. So this mRNA might be present in the cytoplasm. Okay, the whole mechanism, like how the actual mechanism or happens. So I am also a little bit confused about that, but this is what I can, like, the what I explained is what I can deduce from the literature.
Okay, sir.
Okay.
Thank you.
Hmm. So any more doubts? So if not, then we'll move on to the next question. So the shoot apical meristem is maintained by various regulatory genes. Which of the following statements are true for the regulation of SAM activity, that is shoot apical meristem activity? So shoot meristemless gene is expressed in shoot meristem cells and helps in maintaining the cells in their undifferentiated state. Wuschel gene is especially expressed in the organizing center and induces a signal to the overlaying cells to behave as stem cells. And the third one is shoot meristem cells secrete proteins encoded by clavata family that represses Wuschel expression, restricting the size of the shoot meristems. So, can anybody take a guess? What can be the answer?
Option D, sir.
Okay, any more guesses? So yes, option D is the correct answer. And as we can see in this figure, that this is the organizing center, and these four genes are involved in positioning as well as maintaining the stemness or the stem cell nature of the organizing center, such that it will develop into a SAM maintenance. That is, so uh, in case of the shoot meristemless gene, so it helps in maintaining the cells that are in undifferentiated cells. So as you can see from this figure, that when the shoot meristemless gene is knocked out, so the should look differentiated here, and the meristemless gene helps in and it the figure shows that it is without meristem, that is it's in differentiated state. Okay, and in case of the Wushel gene, it is only expressed in the organizing center that's there, and it induces a signal to the overlaying cell to behave as stem cells. So if this region does not have a stem cell, then the whole apical meristem cannot grow. That's why these cotyledons have grown in this case. And the clavata family represses Wushel expression, restricting the size of the shoot meristems. Okay, so this can be seen from the clavata mutants. Okay, so anybody has any doubt in this? Yes, so we are moving on to the next question. So it states that how can we study whether the microRNAs are involved in a particular plant development process? So using only the DCL1 mutant system, first option. Second option is using a DCL1 mutant system combined with specific development gene knockout, then using only specific development gene knockout, or using a Krelok system. Can anybody take a guess?
Option B, sir.
Okay, any more guesses? So yes, the answer is option B. And what happens is this DCL1 is basically a dicer enzyme that is present in plants. So it helps in the formation of microRNAs. And why we are using a specific development gene knockout with the DCL1 mutant? Because if we mutate the plant with the DCL1, then the plant will not be able. When we study any uh, when we study any development process or any signaling process involving microRNA, then we mutate that gene under consideration as well as the DCL1 gene if we have a hypothesis that this region can be involved in this microRNA can be involved in this particular pathway. So we will have both mutations, that is the DCL1 mutations as well as the specific development gene knockout in the same plant. Okay, so anybody has any doubt in this question? Just a second. Yes. So the next question is, there is more amount of fabulosa transcription factor present in metaxylem because the options are high amount of MIR 165/166 present in metaxylem, high amount of auxin present in metaxylem, low amount of MIR present in the metaxylem, or none of the above options is correct. Can anybody take a guess?
Option A, sir.
Okay. Any more guesses? So yes, the correct option is the correct option is option C. Okay. So what happens is, in this case, the scarecrow proteins are the short root proteins are developed in the vascular tissues and are transported in the endodermis. Then there, these short root and scarecrow proteins helps in the formation of the microRNA, and these microRNA move towards the vascular tissues, that is the metaxylem. So it will have more concentration towards the layers that are near to the endodermis, it moves to the nearby cells towards the inside, and it so the regions that are near to the endodermis will have high concentration of this mir. That's a like concentration gradient is formed, and in the metaxylem it will have very low concentration. And as these mirs are like low in metaxylem, these fabulosa transcription factor, that is homeodomain transcription factor, are more in metaxylem as compared to the endodermis. And if we talk about the metaxylem and the protoxylem, so what happens is mir is more in case of protoxylem as compared to the metaxylem, and these transcription factors will be less in protoxylem as compared to the metaxylem. So this is the mechanism that plays the role. So anyone has any doubt in this?
So here, whereas in protoxylem, the activity of miRNA and homodomain transcription factors, there will be an equal amount, so that the activity of that particular fabulosa transcription factor will be altered or will be maintained in equilibrium position.
So as per the concentrations that are present in the protoxylem, that will be maintained at the equilibrium state. Okay, because equal amount is present, so it will show some activity but less than the metaxylem. The transcription factor will show some activity in the protoxylem, but that will be less than the metaxylem, because the concentration of that transcription factor will be less in the protoxylem.
Okay, sir. Thank you, sir.
Okay, any more doubts? If not, then we'll move on to the next question. So, which of the following is an incorrect pair? So, VOX5 division, RBR is to differentiation, SMB is to terminal differentiation, and FES is to differentiation. So, can anybody take a guess?
SMB terminal differentiation.
Okay, any more guesses? So the correct answer is option D, that is phase, that is related to differentiation. So what happens is, in this case, this is the region of the quiescent center and the below the columnella region. So as we can see, in case of quiescent center, there is no division and differentiation. Okay, that's due to these three things, that is oxen, RBR, and VOX5. And in the columnella initials, we will have more division as compared to differentiation. So that's because of VOX5. Okay, so that's why option A is correct. Then, in case of the other columnella cells, what happens is there is differentiation. Okay, so what helps in that is RBR. So RBR helps in the differentiation. So that's why B is correct. And in the terminal cells, so we have terminal differentiation. So these three proteins helps in the terminal differentiation, and one of them is SMB. So option C. And FES here is related to division, not differentiation. So that's why it's an incorrect pair. So option D is the answer. Any doubts?
Says that phase initiates differentiation or divisions, because in previous question we have seen that Phase is also playing a role in columnar cell.
Yes. So what happens is, in the previous case we have seen that in columnar cells, when it divides, then phase initiates the division and it is being moved back into the stem cells from the proteins. So so phase is basically involved in division, not differentiation. Okay, so phase is involved in division. That's why it's present in the stem cells, not in the differentiated cells.
Okay. Thank you, sir.
Okay. Any more doubts? So we'll move on to the next question. So you are studying the sieve tube element development using the glucocorticoid receptor inducible system. You created a mutant with NEN4 and you add NEN4-GR to the roots, and after some time you add dexamethasone to the roots. Now you stain the roots with DAPI to visualize the nucleus inside the sieve tubes. What would be your observation after adding dexamethasone? So the options are: DAPI stains will be present all over the sieve element. So DAPI stains will be present in the initial sieve elements. DAPI will be present in the later sieve elements, or there will be no DAPI stains in the sieve elements. So can anybody take a guess? So the answer will be, sorry, the answer will be B, not A, because first we'll understand the question. So what we are doing is we are creating a NEN4 mutant that helps in nucleus degeneration of sieve elements. Okay, and we are using a GR inducible system out of it. So when we will add dexamethasone, then only NEN4 will be expressed. Okay, so we add dexamethasone and NEN4 will be expressed. Then we add the stain the tissues with DAPI. That's a positively charged. That's a negatively charged dye that stains the nucleus of the cell. So as NEN4 will help in degrading the nucleus, so the DAPI stains will be visible only in the initial stages and initial sieve elements, and in the latter sieve elements the nucleus will be degraded. So this option will be B, not A. This can be better explained with the image. So NEN 1 to 4, we know that these are helping in the nucleus degeneration process, and if we see the wide type, these are the initial sieve elements and these are the later sieve elements that have a nucleus degenerated. Okay, so when NEN4 is mutated in this case, we can see some nucleus present in that, the later sieve elements also, because the nucleus is not fully degenerated. So when we add dexamethasone, so the NEN4 will be present here and it will help in the degeneration of the nucleus. Okay, so the DAPI stains will be like in this case the DAPI stains, DAPI will be blue in color, so it will be visible in the initial sieve elements rather than the later sieve elements. Okay, so any doubts in this? So here we are using a glucocorticoid inducible system in a NEN4 mutant plant, and we are using NEN4 under the GR promoter. So when we add dexamethasone, so NEN4 will be produced. Okay, and we know that in case of sieve elements, the initial sieve elements will have the nucleus, and the later sieve elements will not have the nucleus, because it is degenerated because of the presence of NEN1 to 4 proteins. Okay, so when we add dexamethasone, NEN4 will be expressed, and it will help in the degeneration of the nucleus in the later elements. So now when we stain this tissue with DAPI before as well as after adding dexamethasone, so the before case will be like this, because NEN4 is mutated in this, and it will show stains in the initial as well as the final later sieve elements also, because NEN4 is not there, and the nucleus will be degenerated and will not be degenerated in the sieve elements. Okay, but when we add dexamethasone, NEN4 will be produced, and it will go and degenerate the nucleus. So DAPI stains will only be present in the initial sieve elements, not the later ones. Okay.
Yes, sir. Thank you, sir.
Any more doubts? If not, then we'll be moving to the next question. So what is the function of ACR4 protein in the root development? So it restricts the positioning of VOX5 to QC only. It is present predominantly in plasmodesmata. It negatively regulates the stem cell activity, or both A and C are correct. Can anybody take a guess? So option D is correct. So why? Because VOX5 is present in QC. Okay, so what happens is this clavata 40 controls ACR4, and ACR4 restricts the position of VOX5 to be present in QC only. Okay, and this ACR4 also help to negatively regulate the stem cell activity of this QC in response to clavata 40. And so these are the functions of ACR4, and this is the positioning of ACR4. So it's predominantly present in the plasmodesmata. Okay, so anybody has any doubt in this question? If not, then we'll move to the next question. So considering auxin and cytokinin signaling in roots, which of the following is correct? So auxin signaling predominates in protoxylem, cytokinin signaling predominates in protoxylem, auxin signaling predominates in procampnium, or both auxin and cytokinin signaling predominates in protoxylem. So yes, the answer is A. Auxin signaling predominates in protoxylem. So how did this happen? So auxin controls AH, and all these regions help in the production of cytokinin, and this cytokinin moves to the procambium, and here cytokinin signaling only takes place. So this is how the auxin cytokinin signaling maintenance takes place in the plant. So in different regions, like in protoxylem, majorly auxin signaling predominates, but in procambium the cytokinin signaling predominates. So the correct option is A. Any doubts in this question? If not, then we'll move on to the next question. So, Vox family of transcription factors is a homeodomain containing transcription factors. So, mark the incorrect statement from the following. So, Vox5 helps in positioning of the QC in roots. Okay. Vox2 helps in asymmetric division in shoots. Vox8 or 9 determines the basal fate of endodermis in the roots, or Vox4 stem cell fate in procambium. So can anybody take a guess?
Option C, sir.
Okay, any more guesses? So yes, the correct option is option C. And so first, Vox5 is present in QC in roots and helps in its positioning. Second, in shoots, Vox2 helps in the apical fate of the shoot, as well as Vox8 or 9 helps in the basal fate of the shoot, not the root. So option C is wrong. And Vox4 helps in the to maintain the stem cell fate of the procambium. Okay, so this is how different Vox gene functions. So any doubt in this? If not, then we'll move on to the next question. So you are studying a signaling pathway in plant development. You have to develop a knockout of a specific gene. How can you study that? Or what system can you use? So the options are Krelok system, RNAi technology, CRISPR-Cas9, or all of the above options are correct. Can anybody take a guess?
Option B, sir.
Okay. Any more Any more guesses?
All of the above, sir.
Okay. Any more options? Just a second. So the correct answer is option D, because all of these systems can be used to generate a knockout. So how we look? So first we'll discuss the CRISPR-Cas9, because this we have discussed in the previous lecture also. So CRISPR is basically a sequence that helps in determining the region that needs to be cut, and Cas9 is a endonuclease sequence that is adjacent to PAM and using this we can generate a knockout. uh, Using this we can generate a knockout of a specific gene, to which these sgRNA or the guide RNA is directed. And the second one is Cre lock system. So what happens is Cre is also a recombinase and it cuts only at the sites where lox P sites are present. So if we have to cut a gene, so we generate a mutant that has lox P site around that gene. Okay, so when we add a Cre recombinase to that system, it removes that gene and a lock site, and one lock site is retained at the original position. So this gene Y is knocked out of using this Cre lock system. Okay, and we can use specific promoters to express this Cre recombinase, or the cell specific promoters using which we can express in Cre recombinase in specific cells only. And the third option is RNAi technology. So if we introduce a gene that expresses the miRNA or the siRNA specific to the gene in the plant cell, so this can also be used to knock out a specific gene, that is why in this case we are taking an example. So if we introduce this gene into the genome of the plant that expresses miRNA or siRNA against that gene Y, so this can be knocked out of this plant. So these are the three systems which we can use to generate a knockout. Okay, any doubt, or do I need to explain this question again?
So actually, RNAi technology is used for knockdown purpose only, no, sir. Can we also include, sir?
Okay, so yeah, it can be used for knockdown, and like I said that if we like introduce a gene into the genome of the plant itself that expresses this specific siRNA or miRNA against this gene, then this can be used as a knockout thing.
Okay, sir. Thank you, sir.
Okay, any more doubts?
No, sir.
Okay, so we'll move on to the next question. So, a scientist was studying the polar transport of oxygen by using a DR5 promoter linked to reporter GFP. So, after adding it to the roots, he found that the green signals in the differentiated region of roots shifted to from pericycle to the tip of the roots following some stages. So, after some stages, the the GFP signal or the green signal shifted from the pericycle to the end of the roots. So, what was the scientist studying, or what experiment was he planning? Okay, so the options are vascular development of the plant, lateral root development, shoot cap development, or shoot development. So, can anybody take a guess?
Lateral root developments.
Okay, any more guesses? So the answer is option B, lateral root development. So first we check how the DR5 system works. So as we discussed that oxygen has, has two components signaling, it has two components, IAA and the ARF, oxygen response factor. So when oxygen is not there, it binds to ARF and it is and ARF is not able to function, And when oxygen is there, so ox I use to study the different regions using oxygen as a inducible system. Okay, any doubts? Or shall I explain this again, how this function, how this GR5 promoter functions? If not, then this was the last question for today's session. So if you, any one of you has any doubt from the lectures, can ask. So you can ask from the PPT if there are any doubts, or from the lectures of week 3.
So I have one doubt, sir. What does that RBR means? Is a retinoblastoma, right? So actually, retinoblastoma is only present in eye cells of humans, like rod cells or cone cells. Then how it will be present in plant cells?
So it's basically a retinoblastoma like mutant, like protein that is present in the plants. That is not the same as retinoblastoma protein present in the eyes. So it's just the name given to that, and I also don't know the history of how the name was given to that particular plant. But it's different from the one that is present in animals.
Is it a mutated protein or it's generally naturally present in plants, sir?
It's generally present in the plants because it helps in the scarecrow short root protein signaling. So it's present in the plants.
Okay, sir. Thank you, sir.
Hmm. So any more doubts? So Diusha has raised her hand since a long time. Do you want to ask any question? So if there are no more doubts, we'll be closing the session for today. So thank you everyone for being present.
Thank you, sir.
Hmm. Okay.
Thank you, sir.
Thank you, sir.